Okay, so it's a pleasure to introduce Jack Tuszyński to all of you. He is a colleague and dear friend of mine, and I've been following his work, especially in biophysics, for many years, and have always greatly admired and appreciated his original touch, I must say. And a few years ago, I had the great pleasure to undertake some scientific collaboration with him, and I always found really aspiring to work with him. He has fertile creativity and strikingly vast knowledge, ranging from physics, biophysics, biology, pharmacology, medicine. So, and it is very hard to make a short presentation of Jack, and so I will try to do this in few words. But looking at his CV is really, his CV is truly impressive and reflects his qualities of an outstanding scientist of the highest international caliber. And for example, I've prepared a few notes, okay, of biographical notes. He held visiting professorship and research positions in China, Germany, France, Israel, Denmark, Belgium, and Switzerland. He published almost 540 peer-reviewed journal papers, 12 books. He delivered almost 400 talks at conferences all over the planet, and half of them were invited presentations. He submitted 15 reports of invention, 21 patent applications, and obtained 4 patents in the USA, South Korea, Japan, and Singapore. And his research has been supported by over 100 research grants from Canadian, US, and European funding agencies. He's on the editorial board of almost 30 international journals, including the Journal of Biological Physics. He's an associate editor of the Frontiers collection, Springer Verlag. And okay, he obtained his PhD in condensed matter physics in '83 from the University of Calgary in Canada. From '83 to '88, he was a faculty member at the Department of Physics of the Memorial University of Newfoundland in St. John's. He moved to the University of Alberta in 1988 as an assistant professor. Between 90 and 93, he was associate and then full professor at the Department of Physics. Between 2005 and 2020, he has held the prestigious Aller Chair in Experimental Oncology at the Cross Cancer Institute, where he leads an interdisciplinary computational drug discovery group. And is also a fellow of the National Institute for Nanotechnology in Canada. About his research interests, very quickly, he has been working and still working on computational rational drug design. A lot of his very important work has been concerning microtubule assembly and function, models of motor protein function. He has been working on the integration of mathematical modeling into the pharmaceutical development process. He is an expert of electromagnetic effect on cancer cells and subcellular structures, and on quantum biology. These are only a few notes, and extremely far from being exhaustive. And it's a pleasure to have him with us, and I would leave the stage to him.
Okay, so I guess it's my turn, isn't it?
Yeah.
Well, thank you very much, Nina. Thank you, Marco, and thank you, Jeffrey. I hope you'll see it recorded. It's a great pleasure to be with this excellent group of thinkers deep thinkers. I feel it's my privilege to be part of this community, spanning the globe, as we heard from Nina, all the places mentioned. I'm currently in Turin, Italy, enjoying the nice weather and the historical places I visited, walking from my apartment to the office where I'm right now. And as Marco said, I've been working on rational drug design for the past 15 years. And in the last several years, actually, I was thinking about a new direction because my appointment in oncology ended in 2020, last year. And I think this new direction is what I'll be talking about to you today, namely electromagnetic, electrodynamic interactions with biological matter. And I think we have a lot of interest in common with Marco in this regard, and maybe some of you as well. Let me share the screen now, and I'll go through my presentation, and so then we'll have Q&A. Yeah, this is the title. Basically, it promises something which is not there yet, to uncover electrodynamic design principles of living cells. And I think it's not a very distant goal, in fact, but I think we're not there yet. And in fact, much of it has been hidden from sight, or maybe hiding in plain sight. There's a lot of electrical interactions, electromagnetic interactions, but much of the literature on this right now is kind of spotty and not very well organized. So my interest has been for the past 20 years or so in tubulin and microtubules, and this will be the focus of this talk, but I think the consequences go well beyond microtubules, although they play a very major role in cells. So I want to acknowledge my collaborators, people at the University of Alberta I work with, students and professors, Al Meldrum, Karthik Shankar, Vahid Razania, and the students listed there, as well as my collaborators outside of the University of Alberta in this area, all of whom are in the US at various places. And also, of course, as Marco mentioned, a lot of funding came into it from different sources on different aspects of this project. Actually, the fact that there's a lot of different agencies supporting it, it's probably a negative comment on my achievement, because most of it is in dribs and drabs in small amounts. So rather than a major big grant that would put it on a different orbit, but we are kind of chomping at the bits and trying to carry the research forward. I just want to mention two of these institutions here, which provided funding for this work. One of which is Novocure. It's an Israeli company specializing in electrical therapy for cancer, using electrical fields. I will mention some of the work, not much, but a little bit of this at the end of this talk. The second institution I want to mention is the Allard Foundation. It's a charitable organization which supported my work for 15 years as Allard Chair at the Cross Cancer Institute. And the last one among those that needs special acknowledgement is DARPA, which is everybody knows, the Defense Advanced Research Projects Agency, which recently got very interested in electromagnetic communication between living systems. So that I guess is an acknowledgement of the importance of this area. If the US military is paying attention to it, I think maybe it's time to put some real money into it. So this is the outline of my talk. I will first talk about the tubulin microtubules and their biological structure and function. Then I'll move to some of the electrostatic properties of tubulin microtubules. And then electrophoretic mobility, in other words, movement of these structures under electric fields. And then something which we discovered as, I mean, in retrospect, it's obvious, but I think it's to understand it deeply is not so obvious. The environmental conditions affecting electromagnetic properties of these structures. In biology, nothing is in vacuum, so to speak. So we have to really pay attention to the medium in which these structures exist. So that leads to ionic conduction and capacitance properties, and then the responses to electromagnetic fields at different frequencies, terahertz visible ranges of electromagnetic radiation and different effects that we observe. Much of it is experimental work. I'm a theoretician, I'm a computational scientist, but in the last 20 years, I haven't really worked on things which are not experimental testable. So quite often it's collaborators that do the experiments. In my group also, we did experiments, and there's a spelling mistake. And then we have tryptophan fluorescence microtubules leading us toward these esoteric, I guess, and very attractive quantum properties, perhaps, of biological systems such as microtubules and others. And then the role of pH and all of it. And towards the end, time permitting, I hope it will, I will say a few words about how it all links together into the cellular behavior with respect to electromagnetic interactions, and in particular cancer cells, which I spent about 15 years of my life trying to understand how cancer cells differ from normal cells. And my objective in the path forward is to understand the blueprint for cellular electrodynamics in a more systematic fashion. Okay, so I will start with the basic introduction to microtubules for some of you who are not experts in this area. Microtubules are present in all eukaryotic cells, and tubulin, which is the building block of microtubules, has been on this planet for 2 billion years. So it's a highly conserved protein in all eukaryotic cells and organisms ranging from plants to animals, including humans, of course. And the most highly expressed organs where tubulin can be found is the brain. So our neurons are packed with tubulin and making microtubules in bundles over which a lot of exoplasmic transport takes place. So these are roadways for intracellular transport, and they are also force generating structures. Sorry, in dividing cells. We'll come back to it at the end of the talk when we talk about cancer cells in particular. Together with actin filaments, microtubules make the most important cytoskeletal structures as well as intermediate filaments. But I will spend almost all of my time on tubulin microtubules today. So what's a microtubule? It's a cylindrical polymer composed of tubulin dimers. So it's a heterodimer composed of alpha tubulin and beta tubulin in a high affinity stable structure. You can see here these protrusions on the left of alpha beta tubulin, which are called C termini, which are highly charged. We'll come back to it in a second. Of great importance because they are loaded with electric charges. And of course, this picture doesn't tell the story, but when you look at the charge distribution, you will understand it. And then you see the assembly and disassembly of microtubules, which is a unique biological process. It's a nonlinear dynamic process with stochastic catastrophes. And for some reason, and probably one of the reasons for it is the utility of microtubules in cell division, they have a tendency to grow and shrink intermittently, because for cell division, you need structures to grow and connect to chromosomes via kinetochores, and then you need them to start shrinking and carrying the divided chromosomes to their opposite poles. Now, we'll discuss in the rest of the talk these somewhat exotic still, but to me actually quite commonplace electrical properties of microtubules, which normally I think biologists would not pay attention to, but they are quite important because microtubules can be either conductive or resistive, could be capacitive or not. We also published a paper showing their memristive behavior. That is, they carry currents with memory of the past events. So there is a lot of interesting, non-trivial behavior, to say the least. And then they also respond to electromagnetic stimuli from the environment, showing effects such as delayed luminescence and instability under exposure to visible light, as well as near infrared and terahertz radiation. Some of it, of course, is not present in the biological environment, but nonetheless, knowing these properties is useful for us to reconstruct the physical properties of these biological polymers. This slide shows you the unique properties of tubulin. Quite often I get this question, what's so special about microtubules and tubulin? It's just another protein. Well, no, it's not just another protein. It's actually incredibly multifunctional. The cylindrical structure that microtubules make is unique also, and it is used in supramolecular structures such as centrosomes, cilia and flagella, in cell propulsion, mechanics of the cell, etc. So it's not another protein. And in terms of the electrostatics, is really an outlier. I don't know why this is jumping ahead. So you can see here that the net charge on the tubulin dimer in vacuum is minus 50 elementary charges approximately. It depends on the isotype. I don't want to go too deep into the weeds because this could take another hour easily if I get into some specific details. But suffice it to say that this is a very uniquely negatively charged protein. And then when you introduce counter ions to the solution, the charge, of course, will be neutralized. But the dipole moment of tubulin ranges, again, depending on the isotype, anywhere from 2,000 to even 3,000 dBi. By comparison, a water molecule carries a dipole moment of about 1.8 dBi. So this is massive. Of course, the size of tubulin is much bigger. It's about the dimer is 8 by 5 by 4 nanometers. Microtubules are 25 nanometers across and typically about 10 to 20 microns long. They can be millimeters long under laboratory conditions. So very amazing structures in their own right. This is a computer rendition of the charge distribution of on a microtubule. On the left, you can see here red negative charges and blue positive charges. It's not uniformly negative, but it's interspersed with some positive charges, which can be very important for various functions, such as binding of oppositely charged molecules, including counter ions, as well as things like microtubule associated proteins, MAPs, MAP tau in particular. This again would take me on a different tangent, going into neuroscience, things like Alzheimer's disease and Parkinson's disease, where these things play a role. But what I want to point out here as well is the fact that the cylinder is full of holes. We call them nanopores. So every 4 nanometers or so, you see a 1 nanometer size nanopore through which not only ions, but also some small molecules penetrate into the inside of the microtubule and into the lumen, the space, which is kind of mysterious. Nobody knows what's going on in the lumen, which is 15 nanometers diameter inside of the cylinder. We know for a fact that some drugs such as Taxol, which is used in chemotherapy, penetrate through these nanopores and make their way to the lumen to stabilize microtubules. So with all these charges, there is, so we are now moving into sort of electrodynamic understanding of things happening there. It's not just electrostatics, but also response of charges to electric fields and alternating electric fields, electromagnetic fields. That speaks to the property of polarizability. Polarizability can come as a result of several things. The permanent dipole can reorient itself in electric fields. maybe causing also structural changes in tubulin, conformational changes. The C-termini I mentioned to you carry about 35 to 40% of the charge. They're very flexible, so they are almost like flexible antennas that can respond to the local electric environment. And they also bind motor proteins and maps. So there's a lot of scope for manipulation of these interactions with electromagnetic signals right from the bat. I don't believe anybody has really done any experiments on this, and that's an experiment asking to be done. For example, motor transport under on microtubules under electric or electromagnetic fields. And then finally, there's also the possibility of counter ions surrounding the microtubules to respond to electric and electromagnetic fields, as well as water molecules, which are ordered around hydrophilic surfaces, such as those of microtubules. These are measurements of Aristide Dogariu and his team. I collaborated with them on some interpretation of it, showing the polarizability of microtubules and the dipole moment depending on the local pH. So I will be repeating this message a few times throughout the talk, that there's surrounding is an integral part of the structure. So if you change pH, you change the properties of the microtubule itself. As you can see here, pH affects the net charge because of the protonation states of amino acids. For example, it affects the net dipole. It also affects the polarizability on the left. And there were some experiments done by others on responses of microtubules to external electric fields, such as alignment to electric fields, drift, and effects on polymerization. We are not the first ones, but I think we've done quite a bit more, as I will explain in the rest of the talk. There are several effects that exist in terms of electric field interaction with tubule and microtubules, including realignment, reorientation, and these involve distinct physical mechanisms depending on the intensity and frequency of these fields. Although we have not really mapped it very precisely, but we know that there are effects due to electroosmotic flows of ions, thermal conduction. There's a small amount of heating possible due to these movements of ions and biomolecules in electric fields, and dielectrophoresis, which is basically the movement in the of the dipolar structures in gradients of electric fields. So I would say that there is a whole new planet emerging in front of our eyes in terms of the many dimensions that need to be exploited to find out exactly how this works. And since I told you how important microtubules are for the life of a cell, in terms of transport, in terms of mechanical stability, in terms of cell division, I think this this promises to offer us new possibilities for things like therapies, as I will at the end show you one such existing therapy for cancer with very promising results, maybe some other diseases, neurodegenerative diseases in particular, etc. So frequency-specific responses, we kind of give you here kind of orders of magnitude, what may happen or we have seen happening, but that nothing systematic has been done on building higher level structures from microtubules and how these could be affected by different frequencies. Single microtubules, actually, we have unpublished results I'm not going to show you, can be broken by ultrasound, high frequency ultrasound in around 0.5 to 1 megahertz. There is ionic effects in the 0.1 to 1 megahertz region, conformational changes in the low terahertz range. I will show you that. You can actually destroy microtubules with low frequency terahertz of sufficient intensity, of course. And then there are electronic, excitonic, and visible light frequencies to which microtubules. So this is hundreds of terahertz in the frequency range. Now, centrioles are, as I guess I will not show you, but you see centrioles here on the left. On the left are composed of 9 triplets of microtubules. And 2 centrioles make a centrosome, which is one of the key enigmas of cell biology. It's been dubbed the eye or the brain of the cell, and is very sensitive to light and possibly orients the cell towards the source of light. And again, this is composed of microtubules in a special local environment. So the question arises: what sort of things are conducting? Because as I'll show you, there is conductivity galore. We can have ionic conduction, counter ions around microtubules attracted to it would be able to flow along a direction given by the axis of a microtubule. You can have protonic conduction, basically H plus species, as well as possible electronic. And we've done very preliminary results. I'm not in a position to present them. For dry tubulin, you can measure conductivity of dry protein. And in this case, it would be nothing but electrons that can conduct electricity. And we've seen that it's much less than microtubules in solution, but we'll come to it in a second. So my first statement, not a hypothesis, but it's a statement of fact, that microtubules act as electrically conductive wires, or call them biowires if you wish, as well as capacitors, depending on the conditions. They also store charges, counter ions, but depending on the concentration, so in low ionic concentration, they are vastly more conductive than the solution. At high ionic concentration, they are less conductive than the solution, but highly more capacitive. So the function of this structure can change or can be even controlled with a single knob ionic concentration that the cell has at its disposal. We can also use them for technological applications down the road with this knowledge. So our assumptions are that there are several distinct mechanisms involved in the ionic conduction, one of which is just that microtubules impede flows because they are bulky and they would basically pose barriers, physical barriers to flow. But that's kind of boring. More interesting is that they attract and accelerate ions in favorable directions. And then they also condense some of the ions. According to the Debye theory, you can actually even calculate the amount of condensed ions on microtubules. And then the last one, which is in fact the most intriguing and the least known, is the role of C-termini for creating ionic fluxes. C-termini, these flexible protrusions full of charges, they can basically contribute to ion fluxes. In fact, even inside the microtubule, as we've done through simulations, but experimentally, that's very difficult at this stage to determine. There were a lot of measurements done by various groups on using different equipment, different types of experiments, trying to determine the conductivity of microtubules. As you can see here, the last column gives you the Siemens per meter value of conductivity from various groups, and the numbers vary by 7 orders of magnitude, which tells you that there is something going on here. But keep in mind that different conditions, environmental conditions, and different measurements can probe very vastly different ranges of behavior. So this is my kind of first foray into this field in early 2000s with Horatio Cantiello, who was at Harvard at the time, and Avner Priel from Bar-Ilan University. We did measurements of conductivity or signal propagation, electric signal propagation along a single microtubule. You can see here in the upper right corner two microelectrodes grabbing a single microtubule, as an image that Horatio took of his unique effort in being able to do this. One of the electrodes is injecting the current and the other is collecting it. And as you can see here, the collected current, which is in red, and over 5 milliseconds, it's a signal with 5 millisecond duration. The collected current is actually amplified anywhere from 2-fold to 3-fold. So microtubules in this case act as amplifiers of signals. And we published this in Biophysical Journal, that this was very intriguing. And we created a model of microtubules as RLC devices. So each tubulin unit actually is an RLC circuit, and you create a network of these circuits representing the entire microtubule. These are the values that we obtained from estimates and published that result. And based on this approximation, we've created continuous model for the resultant voltage propagation along the direction of microtubule, which is the x-axis here. It's a very complicated nonlinear differential equation. Amazingly, we solved it exactly by introducing moving coordinates and simplifying it, and you obtain something along the lines of a damp-driven nonlinear harmonic oscillator, leading to elliptic waves and solitary waves, which would explain some of the observations at least. And that was extended to a sort of three-dimensional representation when you also allow flows inside the lumen and outside. Okay, what's interesting about this, I will just point out one interesting property of this, and it's actually experimentally verifiable, that the microtubule wall resembles a cellular membrane in some respect. The thickness is similar, about 5 nanometers thick. It has also nanopores, just like membranes of ion channels. And the potential difference between the outside and the inside of a microtubule is about 100 millivolts, which is also comparable to membranes in cells. Therefore, one can think of this as maybe some sort of proto-membrane, maybe something in a simple organism, or even prior to the formation of full-blown organism. Microtubules could have been formed by self-organization in some sort of early primordial soup. But okay, so this is one slide which shows you where other charges reside in living cells, a lot of them. You have a preponderance of sodium outside in extracellular space matrix, potassium inside with the concentrations given in the table. Then you have chlorine and calcium, magnesium, much smaller concentrations, and of course actin and tubulin being the most prominently expressed proteins forming these cytoskeletal filaments. Okay, so that's, and the axis on the left with the transmembrane potential is important. I will come back to it. Mike Levin, I think Mike Levin spoke in this series last year, and maybe he talked about it, but I'm trying to connect the dots. This is one of the dots. The membrane potential changes as you go in from normal cells to cancerous cells, as you can, and he probably talked about it a lot. The transmembrane potential reduction to something like 30 millivolts or 20 millivolts, even less, has consequences for ionic concentrations. And I'll come back to it later. So for now, let me go through the remaining recent experiments. Now we're talking about the period from 2017 to today. And these are images of different we call them meshworks of microtubules under experimental conditions, different concentrations of tubulin given here, and measuring their electrical properties under different ionic concentrations. So these are the devices we used: Keithley semiconductor characterization device, and 4-point probes and 2-point probes were used in different experiments. And these are basically the results which we published recently. As a result, as a function of frequency of the electric field, you obtain conductance which peaks at around 110 kilohertz, which is worth noting because that's around the frequency the company Novocure uses for therapeutic applications for brain cancer. It's not a very sharp peak, but we are talking about, the order of magnitude frequency at which conductivity at low ionic concentrations peak. Microtubules were more conductive than tubulin. Tubulin was more conductive than the solution itself. And this is the paper that was published on these results. Then we moved on to measure capacitive properties of ionic solutions in microtubule networks. This is the publication in 2020, where we showed virtually the opposite at high concentrations of ions, actual physiological concentrations, which are somewhere in the 100 millimolar concentration range. And the imaginary impedance, that means capacitance. Here, microtubules are shown to have a greater capacitance than free tubulin, and this is an excess over a solution itself. So, so it's opposite to conductance. And here, resistance, now resistance of microtubules is greater than tubulin, greater than solution, and all of it is frequency dependent to some degree. So at low ionic concentrations, microtubules decrease solutions impedance or increase conductance. In other words, would provide preferential pathways for electrical signals to go through when the ionic concentrations are low. We'll come back to the question of what is low and what is high. But for now, let's say just low, microtubules conduct better than the rest. High, the opposite takes place, but microtubules then become very good capacitors. They don't allow flows of ions so easily, but they do want to store charges. So you can maybe regulate this. You can change their storage versus propagation properties by turning one knob only, namely the concentration of potassium in the solution. If it's a cytoplasm, the cell could do it. So we've calculated this by using this very simple model. And it's really very, very nice, fits into the experimental data. And this is the table that kind of summarizes the results of different tubulin concentrations on the left, with the capacitance of the entire system and the errors in the measurements and resistance. The difficulty of extrapolating this to a single microtubule is real, because these are not parallel or series circuits, and you know how complicated it gets using Kirchhoff's laws for very convoluted systems of resistors and capacitors. Nonetheless, we've done for the lowest concentration some simple extrapolation, and we got values which are roughly, if you look back to some of the earlier slides from 2006 modeling, these values are the same order of magnitude. So they are consistent with these, 15-year-old computational predictions. And this is, so that's comparison between experiment and theory. We're in the same ballpark. But remember, all of this is frequency dependent, is concentration dependent, and also scales with length in different. So resistors scale linearly with length, capacitors too, inductors inversely. There's also an inductive part, and you might ask me, maybe somebody will ask me, where's the inductor? So we talked about R and C, where's L? Well, L is a solenoidal flow of ions because microtubules have an inherent helical symmetry. It looks like a cylinder, but actually it's a helical helical string woven on the cylinder. So there's a natural pathway along this helical geometry for ions to flow along the grooves of the microtubule. Okay, so then we moved on to verify some of this through different measurements, namely electrophoretic mobility, so movements of microtubules, and tubulin in electric fields. We changed the polarity of these electric fields. You see some very violent movements when you change the polarity. That testifies to the amount of charge stored on them. Now, and then of course it all, and this is a lot of data here, which I probably don't have enough time to explain, but it's all published. What I want to say, some of the most intriguing things that emerged is so now, I told you about the importance of ions. Now there's the second player in this game, pH, so protons. And it so happens that at pH about 5, tubulin becomes positively charged. And so you can now reverse the electrophoretic mobility. If you use microtubules as let's say, building blocks of something you want to construct and drag them to the opposite electrode, then by changing pH below pH 5, now you have, instead of negative microtubules, you have positively charged microtubules. So that's what I mean by this complex planet. It really is multi-dimensional puzzle that we're trying to put together. Okay, and so it also depends on the solvent. So these are different solvents we used, and again, the same conclusion for different solvents at different pH. Around pH 5, you see the electrophoretic mobility changing sign, indicating that, and that's also shown by the zeta potential, that tubulin becomes positively charged. And also at very high, DMSO is a solvent very commonly used in cell biology experiments. For poorly solubilized ligands, use DMSO. For example, in cytotoxic experiments for cancer cells, and use some chemotherapy drugs, put them in DMSO and then apply that to the cell. So at high percentage of DMSO, we also found the same thing. I don't have an explanation for this. That's kind of bizarre, but tubulin becomes also positively charged at high DMSO concentrations. All right, I will skip through this, and now I'll show you some recent simulations explaining this bizarre fact of microtubules becoming capacitive for high ionic concentrations and being conductive at low. So this has to do with the Poisson-Boltzmann equation for solving the distribution of ionic species around a charged surface, such as the surface of a protein or protein filament like actin or microtubules. And so these are the various modifications that account for the Poisson-Boltzmann equation shown in one, and then accounting for variable dielectric constant, and finally for ionic size modification. So a little bit of technical detail. I don't want to get too wrapped up in math, but when you solve these equations, you get some very interesting results, showing you that these are the profiles of potassium, which is the dominant ion concentration around different surfaces of microtubules, the outer surface. So 12.5 means 12.5 nanometer radius from the center of a microtubule, which is basically the surface. And then you see how rapidly the potassium concentration drops over 2.5 nanometers from the surface. For C-termini, you also see this is the radial position of C-termini over 1 nanometer, 2 nanometers drop. And then we solved it for variable ionic concentrations, and that shows you the different profiles of potassium concentration around the lumen, the C-termini, and the bulk. And this is the punchline. When you use these results to calculate the conductivity, you obtain the graph on the right, which is, which is showing a crossover. The red line is the conductivity of the bulk solution as a function of its ionic concentration. It's a straight line. It's so-called Kohlrausch law for, it's like Ohm's law for ionic solutions, straight line. But for microtubules, because there's a different Poisson-Boltzmann type of solution to this problem, giving you nonlinear behavior around it, you see here that conductivity is higher. That's the green line. Blue line is microtubules with no C-termini. So if you remove the C-termini from microtubules, which you can do biochemically, then you have a slightly lower conductivity. But compare the green line with the red line, and you can see here at 100 millimolar or so, there's a crossover. You have the same conductivity of microtubules as for the solution in which they are contained. So that means anytime the solution is below 100, microtubules would be the preferential conduits for ionic movement. Anything above 100 would be the opposite. They would just store excess ions, but would not preferentially conduct. So that's kind of intriguing. And the fact that it's around 100 is worth noting, because this is typically kind of 90 millivolts to 120 normal cells. You recall Mike Levin's talk. If you go into cancer cells, you go into the low 10, 20 millivolts, and the gap widens. The lower the ionic concentration, the bigger the gap. Okay, this may be, in the interest of time, I'll very quickly skip through this. These are recent, just about published results of demonstration on the resolution of 1 to 2 nanometer scale of the fact that microtubules attract positive ions, in this case protons. So pH changes around microtubules have been measured and shown here. You can calibrate them with the wavelength of emitted light compared to the background, and we use fluorescent molecules called P4VB, which bind to microtubules and report. These are basically reporters of the local concentration of pH, and that shows you how this pH differs between microtubules and the solution around anywhere from 6.5 to 8 and higher values of pH. The gap between the solution and microtubules grows, which means that microtubule become more and more of a attractant to protons, protonic species. Okay, now we in the last few, I don't know, maybe not minutes, but I don't know, 10 minutes, is it okay?
Yeah, that's fine.
So in the last 10 minutes, I will now focus on electromagnetic interactions. These were all electric interactions, not electromagnetic. And this is a diagram from a paper of my colleague. Maybe he also spoke here, Michał Cifra from Prague. Yes. And they've done simulations of microtubules as kind of resonators, and this resonant electromagnetic effect was predicted in this graph to be length dependent. So very long microtubules with very low frequencies, and this goes into 100 gigahertz, almost, yeah, so 0.1 terahertz. And so I'm going to start there. These are the experiments that I participated in with my colleagues from Department of Physics at the U of A, Frank Hegman and Cam Howe, his PhD student, Dr. Howe. And terahertz oscillations are kind of mysterious. Marco Pettini has spent some time understanding this and writing about it and doing experiments. We have specifically tried to understand if terahertz radiation affects tubulin stability in microtubules, and we found actually dramatic effects of low terahertz radiation on stability of microtubules. You see here on the left images of microtubule bundles, similar to those that I used in the previous slides for measurements of conductivity capacitance. With terahertz on, they disappear. You can see here images of broken microtubules into pieces at the bottom corner on the right. And this is a movie showing you a time lapse over 15 minutes, how fluorescently labeled microtubules disappear under the influence of not alcohol, but terahertz radiation. So that's intriguing. And what's even more intriguing, I think, is that we tried only 3 different frequencies, and the intensity threshold is high. It's about 500 kilovolts per centimeter of the intensity that is needed to do the job locally. So you have to focus it. But what's intriguing is that counterintuitively, so lower frequency, 0.5 terahertz, has a greater effect on microtubule distraction than higher frequency. And as you know from Planck's equation, E equals h times f, the energy in the higher frequency would be higher, but it doesn't do a greater damage. In fact, the opposite is true. So at about 0.5 terahertz, we see, well, even bigger damage at broadband. So it's a mixture of different frequencies. But 0.5 for a single frequency value seems to be the best. Now we did some simulations with Michał Cifra and my Italian colleagues, and these simulations of electric field effects on the structure of tubulin, not whole microtubules, but structure, kind of are consistent because they give us similar intensity value for permanent conformational changes in tubulin. Some movement of helices, movement of C-termini, and reorientation of the permanent dipole, which would make tubulin very uncomfortable being in a microtubule. And I think this probably is an explanation for the effect of microtubule disappearance, making basically breaking hydrogen bonds between neighboring dimers and gradually disassembling under the effect of the exposure to electromagnetic field in the terahertz range. We've seen similar effects with other ranges, but here is kind of a table that explains the microtubule stability, which comes from a lot of different contributions, including predominantly hydrogen bonds, but also hydrophobic effects, as well as dipole-dipole interactions and so on. So now on to even more exotic stuff. This is the work of my collaborator, Dr. Aristide Degariou from University of Central Florida in Orlando, where he exposed microtubules in solution to repeated cycles of exposure to visible light in the 600 nanometer, 600, I don't remember. It's not shown here. Over, oh yeah, it is 650 nanometer, yes, a nanometer, which is 1.8 electron volts. For those of you who are thinking in electron volts, that's and the intensities are given. The electric field intensity is actually quite low, 200 volts per meter. But exposure 4 minutes, then there is a gap of 10 minutes and so on, repeated a number of times. And here you see, and then they measure the photon re-emission after this exposure, and for tubulin in solution versus microtubule in solutions, which are in blue, and microtubules seem to be capturing that energy for longer. They re-emit, but both systems re-emit this over a very, very long period of time, on the order of several minutes, which is bizarre to say the least. You would expect something to be re-emitted more or less instantaneously, you know, on the order of nanoseconds perhaps, or shorter. And that's been done for several different situations with aligned microtubules and randomly distributed, and photon counting is given here, and so on. And so so now we are exploring something which brings us closer to this quantum biology theme of this series. So far, everything was pretty much classical, although delayed luminescence may not be classically explained. Maybe there is some sort of quantum transitions there. But here we are talking about tryptophan fluorescence in microtubules. This is done by, maybe I'll just skip some of the theory because in the interest of time. But we published a number of papers with my former student, now professor at Nova Southeastern University in Florida, Travis Craddock, predicting that tryptophan, so this is the network of tryptophan amino acids in a tubulin dimer on the left, and you see 8 of them within something like between 1.5 and maybe 2.5 nanometer distances between them. And that network, if anybody has been talking here about photosynthesis, quantum biology, then you may kind of see similarities between FMO systems and this one. So we did some calculations and published that and predicted that there will be long-lived collective excitations of these tryptophans if they are excited by laser. Okay, and now we are doing experiments on this. We meaning Greg Scholes at Princeton and Narat Kalra, and that's basically the current experiment that we have results, but I'm not at liberty to show you the results. I'm sorry. And we use tryptophan fluorescence using this kind of equipment. Time correlated single photon counting is used, and these characteristic times T1, T2, and T3 are measured. And I can only tell you that we see the emission of these photons from microtubules on the order of between 3 and 5 nanoseconds, which is longer than for photosynthetic systems like, you know, FMO systems in chlorophyll. So that's very intriguing, because that would speak to the possibility of quantum effects and really quantum effects, collective effects in microtubules. Arat is doing experiments with different distances between clusters of tryptophans, and we see dependence on the distance dependence. So it's a collective effect. It's not like a single photon absorption emission. It's relatively long range communication between them. Okay. How much time do I have? Because I can now jump to the
you're okay for 5 minutes.
For 5 minutes, okay.
Or up to 10, yeah. Just so we've got half an hour for questions at the end.
All right, yes. So I will skip through this because this is sort of along the same lines, shining light on microtubules and measuring the polymerization rates. In this case, electromagnetic radiation in the visible range with these intensities slows down polymerization and makes fewer microtubules in the end. So electromagnetic effects do interact. We've seen for terahertz, we destroy microtubules. Here we don't destroy them, but we slow down the polymerization. There's also hysteresis effects in microtubule polymerization and depolymerization processes. And now let's just jump to something which kind of connects my two lines of research, oncology and electromagnetic effects in biology. So microtubules, as I mentioned earlier, play a pivotal role in cell division. You can see them in green here in these images of dividing cells, very beautifully symmetric blue DNA in chromosomes being segregated by microtubules. In other words, you cannot divide a cell without microtubules. So anything that you do to mess up with microtubules in dividing cells, you will achieve potentially an amazing benefit of slowing down, if not stopping cancer. The depolymerization of microtubules in dividing cells is a fascinating mechanical mechanism. You can see here that there's kind of on the left, there's a ring of other proteins, I forget what they're called, and then they are connected by some kind of harness to the kinetochore. And so I always wondered about where the force is coming for pulling the chromosomes apart, which is a very large force, about 750 piconewtons, which is very large. And it turns out that this is a purely mechanical force you see these ram's horns of protofilaments curving from microtubules as they depolymerize. They exert a force on that ring, and the ring slides down and down. You can imagine this as a mechanical device. If you put exert a pushing force on the ring, it will slide down towards the opposite pole, and the genetic material slides together with it. Okay, so now some more movies about why microtubules are also important in other things. And you see here, these are mitochondria actually moving in green in non-dividing cells, and in red here in this corner, mitochondria in dividing cells. So mitochondria actually move on microtubules as well. So you see microtubules everywhere. Maybe that's my sort of aberration or myopia. Okay, and this is a single slide that kind of tells a very important story that I don't think anybody has told before. Maybe Mike Levin, he might be the only guy who did this. So differences between normal cells and cancer cells. You can see here on the left in the upper corner, internal pH changes. So normal cells have lower pH inside, cancer cells higher. So they are more basic inside, more acidic outside. Then you see mitochondrial pH in cancer cells is higher. Mitochondrial transmembrane potential is higher. And then the transmembrane potential is vastly lower. That's the same graph that I showed you earlier. Now it's in black and white and upside down. And that translates into the ionic potential, ionic concentration. So on the x-axis, on the graph with the blue curve, you see the drop in the transmembrane potential means higher potassium concentration. So all of this is linked together. Whatever happens to the membrane happens to the ions. Whatever happens to the ions happens to microtubules. Whatever happens to pH also affects microtubules. I haven't solved all these equations put together yet, but this is that ultimate challenge. And it's been observed that actually that cancer cells also abolish the pH gradient. So in normal cells, there is a gradient of pH from between the cytoplasm and the nucleus. In cancer cells, it's uniform. And part of this, I think, is explains this glycolytic switch, the so-called Warburg effect, meaning that cancer cells become glycolytic, produce lactate, inefficient energy production, very hungry, very aggressive, because they cannot utilize mitochondrial activity. All right. So another connection, right? Mitochondria. And now 2 more treating fields. So this is the technology that started with this paper in PNAS in 2007. So it's 14 years ago, a single paper. Now it's a company worth a few $1 billion in Haifa, Israel. But more importantly, thousands of patients with brain cancer who are gaining at least a year of life without side effects. And again, the question is, what is this electric field doing? And this shows here, that's another paper from Michal Cifra predicting the electric fields around microtubules and dividing cells. You can see how focused this field is around microtubules. And now imagine electric fields from the outside interacting with these electric fields inside. Okay, and here is the experiment, not published yet, experimental data from our measurements. We used Novocure's device called InnoVitro, and these are HeLa cells, or ovarian cancer cells. On the left is normal division of these cells without electric fields. Okay, very nice, symmetrical, pretty fast. And on the right is in the presence of electric fields. You see, they cannot really separate. They are still connected by microtubules, these green things. And we have actually, we have beyond observations, we have measurements showing that these fields slow down the cell division rate by up to 5 times compared to control. So it's very dramatic in terms of the number of dividing cells, and also about 1/3 of the cells die without being able to complete this process. 1/3 become sort of quiescent, and one third divide, but the effects are dramatic. Now the question is optimizing all of this, you see. So my message, I think, and I'm coming to the end now, really, is this. That's the ultimate objective. As physicists and engineers, perhaps some of us, I would like to be able to construct such a simple circuit where we have the membrane as a leaky capacitor, then we have the cytoplasm as a resistor and capacitor, then we have the microtubules in the cytoplasm and maybe actin filaments with equivalent circuit elements, all of this being the cell. Of course, yeah, and the signals being the voltage, ionic concentration, and maybe HF means photons, because these elements interact with all of the above. So how do we, first of all, construct a circuit like that? Secondly, how do we utilize it for whether it be, cancer treatment or some kind of device or prosthetic applications? Different cells will have different parameters, for sure. Okay, so I'm now down to the conclusions. I hope that I've convinced you that there is an amazing richness of behaviors vis-a-vis electromagnetic fields of microtubules, that they can be seen as bioelectric circuits that are sensitive to the environment, which is much more complicated than typical electric circuits, and that they also interact with electromagnetic fields. And actually, we can even control them by electromagnetic fields. As you've seen with terahertz, we could also destroy them at will. So whether you think about nanotechnology or biotechnology or therapy, I think this is a new world that is opening up in front of our eyes. So thank you very much for your attention, and I'll be happy to discuss any of that.